your ideas. What you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Thank you for joining me.
My pleasure.
Do you want to just start off by telling everyone a little bit about yourself and what your lab studies?
Sure. I'm a professor at Weill Cornell Medicine, which is the medical school of Cornell University in New York City. And for the last couple decades, I've been studying the molecular basis of how synapses work, which are the points of communication between brain cells. And with the particular emphasis, I would say, in the last dozen or so years on understanding the roles of local metabolism and bioenergetics and how that shapes how well synapses work.
Yeah, let's give people a sort of a crash course in synapse structure and function. So a synapse is the place where one neuron communicates with another. So you've got a postsynaptic and a presynaptic neuron, right? There's a sort of an input and an output side. What are the major components of a synapse? And I'll sort of let you direct our attention to either the pre or postsynaptic side in terms of where your research focuses are.
Yeah, so just as you said, it's the major point of communication. So to someone who's not used to thinking about synapses, the brain is all about communicating between cells, and the neurons are famous for having the ability to send signals electrically over long distances. But then the synapse is this relay point which converts an electrical signal briefly into a chemical signal that then gets turned back into an electrical signal. So there's this conversion point. Now, how do neurons do this? They make use of a specialized machine called the synapse. Our brains contain probably on the order of trillions of synapses, maybe 10 trillion. And each one is endowed with packages of neurotransmitter. They're stored in small things called synaptic vesicles. They're small, meaning diameters of about 40 nanometers. And each synapse, I would say, even though the numbers can vary enormously, something like 100 vesicles per synapse is sort of the canonical number we walk around with, thinking that's what we should deal with. But it can vary by orders of magnitude in either direction. Mm-hmm. And so when the electrical signal travels down the axon, that electrical signal causes changes in the voltage difference across the membrane. And one of the things that happens is it triggers the opening of voltage-sensitive calcium channels to then allow calcium to transiently come into the nerve terminal. And that is the essential trigger for causing a synaptic vesicle to then merge with the plasma membrane through a process called exocytosis and release the contents of that vesicle, which in this case might be a few thousand molecules of glutamate in the case of the major excitatory neurotransmitter in the brain. Mm-hmm. But what my lab sort of really for 3 decades got interested in was not so much only the process of what's called exocytosis, but each synapse only has on the order of 100 of these vesicles. And it would be inconvenient if every time you use one for exocytosis, it would be done. And so the synapse also has embedded in it an efficient recycling system, to rebuild the synaptic vesicle after fusion by recapturing the components that made up the vesicle from the plasma membrane through a process called endocytosis, and then refilling the vesicle with neurotransmitter so it's ready to be used again. Mm-hmm. That whole process we call synaptic vesicle recycling.
Mm-hmm. So synapses are points of contact between neurons. There are a lot of neurons in the brain. In our brain, we've got billions of them, and we've got trillions of connections. So there are, depending on the neuron, depending on the brain region, thousands and thousands connections per neuron. So you've got a lot of these connection points. They're critical functionally because they're doing this chemical to electrical conversion, whereby an electrical signal causes vesicles, which are essentially little bubbles of fat, to merge and then release chemicals into the synapse. And, that's all just part of how this chemical communication happens, electrical to chemical communication happens. If we start to think about it, in the human brain, trillions of connections, so that's trillions of synapses. Each one's got probably 100, with a lot of variability between synapses, but hundreds maybe of vesicles filled with chemicals in each synapse. You've got all kinds of proteins and infrastructure in this little machine. If you start to think about that, plus the fact that the synapses are often located very, very far from the nucleus of the cell, because neurons have this kind of weird shape, it starts to bring up a lot of questions around, like, how is all this stuff powered and how is energy getting there to do this? Before we dive into the energetics per se, let's just give people a basic idea of some of the key pieces of infrastructure within the synapse. Let's start with vesicles. You mentioned those. What exactly are vesicles?
So synaptic vesicles are bounded by a phospholipid membrane, a bilayer. This is, as you said, it's fat because it has a carbon chain that's usually, maybe a dozen to 20 carbons long, depending on sort of now details, with a charged head group, the top of it. So these things will form this little container that's bounded by something very hydrophobic that doesn't like water and forms a barrier that you then can package in whatever you want. But to package things in the vesicle, you need specialized machinery. So living on the surface of the vesicle are many other types of proteins. In this case, highly relevant to the conversation we're going to have is you need the thing that's going to allow you to put neurotransmitter in it. These are transporters. There's a different flavor of transporter for every different flavor of neurotransmitter. There's one that's specialized for glutamate. There's another one that's specialized for GABA, the major inhibitory neurotransmitter. Another one for acetylcholine, the major neurotransmitter, for example, for allowing muscles to contract, but also present in the brain. Another specialized one for dopamine. And so each one, they're specialized machinery. They're kind of all similar to each other, but they have their own little specialty of why they work well for one type of molecule, not another. And then, but those, so a little thing to think about, a vesicle that's ready to go might have as much as a few thousand molecules of, say, the neurotransmitter in it. Now, the concentration of neurotransmitter in the outside of the vesicle is actually on the order of sort of, let's say, millimolar, for example. But inside the vesicle, once you packaged a few thousand copies, it's probably more like 100 millimolar. So that's a giant gradient, and it takes energy to overcome and build that gradient.
Yeah, because if you concentrate something in a little bubble, it will naturally want to go back out down that concentration gradient. So you have to pump, you have to use energy to keep things concentrated.
Yeah. And one of the big, I would say, discoveries of the last 30 years, say, is the beginning to understand what are the machineries that do this pumping and what do they rely on. And you might have said, oh, well, you can make a pump that's powered directly by something like ATP, but in fact, that's not the way it works. Instead, all synaptic vesicles rely on the action of another pump, a proton pump. That is actually called a V-type ATP, is V standing for vesicle or vesicular. And those live in more places than just synaptic vesicles, but their big thing is they use up ATP to pump protons. And so it turns out the way you do all of neurotransmitter pumping into vesicles is sort of a coupling of the energy you get from pumping protons and creating that gradient first. And that ends up being the power to put in neurotransmitter.
So vesicles contain neurotransmitters, which are key components of synapses used for communication purposes. But vesicles are not merely little bubbles of fat, little bags of fat that contain chemicals. They have embedded in their membrane, just like the main cell membrane of the cell, a bunch of proteins that are important for actually keeping the vesicles filled with whatever chemical a particular vesicle has.
As well, of course, is the whole business end of how do you know when to fuse with the membrane to release your content. There's obviously key protein machinery about mediating how fusion can happen, how you sense that calcium went up. And there's still plenty of mysteries being worked out. But those those things we know have to happen.
Yep. So there are a bunch of proteins in the vesicles, in the cell membrane, within the cell, at the synaptic, at the synapse. These things act as sensors of stuff. These things act as pumps. Like, they are really little machines, and they need to be powered by energy. So there's a big concentration of stuff, and all of its functions don't come for free. A lot of energy is required to make all this stuff happen.
Right.
And so I would imagine this is a big reason, not the only reason, but a big reason why brain tissue is so energetically expensive.
Yes. That's absolutely true. I mean, as you said, there are trillions of these synapses, and the complexity of the synapse means that there are these burdens that you've got to pay the price somewhere. And when you just scale up that sheer number, that probably adds up to a lot.
And so, when we think about cell biology, there's many different parts of the cell. One thing that's interesting about neurons is their anatomy. They're usually long and slender, right? The axon can be very long. Sometimes, it can basically go from one end of the body to almost the other end. And so a synapse might be located very, very far from the nucleus, very far from the main bag of cytosol that surrounds the nucleus, where I imagine a lot of the stuff of the cell is, the ribosomes, the mitochondria that make energy, and so forth. And so this brings us to the question of, well, how do you get all of the fuel and energy and structural, infrastructure support you need to power that synapse to the synapse? Are you shuttling ATP there? Are you shuttling mitochondria there? Or is there local support, like mitochondria right in the synapse and these types of things? When we start to think about the energetics of it and what's powering that, how does a lot of that infrastructure actually get to the synapse?
Right. So let me just back up. So neurons are fascinating. They differ from most other cells in our body for two big reasons, one of which is their architecture, that they span enormous distances, fairly unparalleled in the body. There's nothing else that covers quite as much ground, as you said. The other, of course, is largely neurons are non-dividing. There are small populations still debated about how relevant those are, but it is a minority if they exist in the human brain, for example, at all. And so you're not doing what a lot of cells typically have to do, which is to renew themselves. Now, in addition, you do have to get stuff far away. Right. So, we know that the major things that do transport, like there's specialized motors, but we know their speeds, and that sets sort of times of how long it would take to get something to go millimeters, centimeters, meters, right?
Right.
And that means that the needs we have for powering the brain, of course, are moment to moment. We're not waiting for the shipping company to send us resources, right? We do rely on the shipping company to send machineries and things, but that operates on a much longer time scale. The worry is that you need things moment to moment all the time, and so there you have to deal with it, and it's a local problem. And so the local problem is that the fuel largely, well, there's sets of fuels. We have the machinery to turn carbon sources into ATP. Now, where do the carbon sources come from? I would say the thing we think we understand the most is glucose, because glucose is delivered through capillaries, and the brain has an enormously complex vasculature, and so those distances, you're never far away from where glucose is being delivered, basically. I mean, you may be a little too far away, but it's nothing like the length scales that we just mentioned of, how far a cell body is away from a synapse. Yeah. So at a very high level, taking a bird's eye view here, when we start thinking about the energetics here, it's like, okay, synapses are really expensive. There's many, many, many synapses. We need energy, but neurons are kind of special in that most of the time aren't dividing. And so cell division is very energetically expensive. So right out of the gate, we might think, okay, all of the energy that other cells would be devoting to cell division now can be used for other things. And maybe that has something to do with why neurons got the way they are to begin with. But then there's this problem of distance. And so we've started to say that, okay, we need local energy sources at the synapse. The synapse might be very far from the nucleus, but the brain has a very, very complex network of vasculature that can deliver things like glucose, to many, many different points in the brain. So you're probably never that far from an energy source. When we start to think about things like, okay, if you're not that far away from glucose or a fatty acid or something that can be turned into ATP,
because it can get there through the vasculature and things like that. Well, it still needs to be converted into energy by something like the mitochondria. So are there mitochondria inside of synapses? Do they go all the way up into that
Yes.
part of the neuron?
Yes, I'd say there's two pieces to this. Glucose, of course, gets converted, ultimately can be used by mitochondria, but it actually first has to get converted into something the mitochondria can use, and that's a process called glycolysis.
Mm-hmm.
And that is also a fascinating, complex piece of machinery. It's an assembly of 10 enzymes that need to do this conversion. We discovered a few years ago, and we were not the first, but it sort of hit home with us, that nerve terminals are quite enriched in the machinery for glycolysis as well. But in addition, they typically have mitochondria. I say typically because that is also quite variable. So mitochondria are easily recognized, for example, in electron microscopy. They have a characteristic shape. And famous pictures, for example, if you looked at famous synapses, which are, say, the ones that allow you to contract your muscle, right, they are jam-packed with mitochondria. But if you look in the brain at where there's synapses not between a neuron and a muscle, but between two different neurons, it's considerably sparser. So the average number in most of the literature is about half of synapses have a nearby mitochondria, but only
half of synapses in the brain tend to have one nearby.
One, yeah. Oh, no, yeah, only one nearby. Half of them. So half don't. Half have zero.
Yeah.
So we've been intrigued about the significance of that rather low number. We haven't come to any clear conclusion so far about the significance of that low number. I have a hand-waving explanation for why it may not be as interesting as it sounds, which is that you probably can steal from a neighbor when you need to.
Yeah.
And that depends on how far away you are from your neighbors.
Yeah.
So, and the density of synapses in an axon in the brain is actually relatively high. So you may never be that far away from a neighbor who happens to have a mitochondria.
Yeah. I see. Yeah. So even though a mitochondria might not be in a synapse and it's far away in terms of the line you might trace on the inside of the cell from that synapse to a mitochondrion, there's like lateral movement from one synapse to another, and so maybe there could be ways.
We're still working on seeing if what I just said really holds up.
Yeah. Okay, but all neurons are going to have mitochondria in them. Any given synapse may or may not have one. On average, you said, about half the synapses don't really have one nearby, whatever exactly nearby means, and half of them do. So adjacent to the synapse or in the synapse. And then, of course, there are some synapses like the neuromuscular junction synapses that have lots of mitochondria in them. Yes. So there's a huge spread here.
Yep.
And, just thinking about this on an intuitive level, it makes a lot of sense to me that the neuromuscular junction, that type of synapse, would have a lot of mitochondria, because I would imagine that's naturally and especially energy intensive synapse, because that's what actually where the rubber meets the road and the muscles move to propel the body through space. That's in large part what I think nervous systems are doing, right? They're about moving the organism. Other neurons in the brain do very different things, and there's lots of diversity. At a high level, is there any obvious mapping between the mitochondrial density at a synapse and like what it's doing in terms of its energy intensity, or is it all kind of mysterious still?
I would say it's mostly mysterious. What seems interesting is still mysterious. So there are synapses in the brain which also have extremely high density. I'll give you an example.
Yeah.
It was a model that was sort of famous amongst biophysicists for the last 30 years because it was a giant relay synapse in the auditory system. And its job was really to relay auditory information, not so much processing it, right? It's just like in the chain of command going from a sound causing a vibration in your ear, there are these synapses, and a famous one was in the auditory brainstem called the calyx of Held. Biophysicists loved it because it was big, you could patch onto it, make recordings, be as reductionist as you could. Those ones famously are jam full of mitochondria as well. But I view them as sort of the equivalent of the neuromuscular junction in their job. It's sort of a fail-safe, pass it through, do this really well. And so resources have been developed to make sure that doesn't fail at the energetic level, I presume. Now, we don't understand what there when you cast through most of the rest of the brain aren't synapses that look like that, right? They're these small, tiny little varicosities that may or may not have a mitochondria.
Right.
And so that's where I would say there's still plenty of work and discoveries to be made about the significance of the density of those and the details. I mean, I'm optimistic we'll begin to learn those things as better tools become available.
And so when we think about just a synapse, I'll let you pick the examples, but when we think of just a canonical synapse, what are there particular components of the synaptic machinery that are sucking up most of the energy, or is it kind of spread across lots of different things? I just want to get thinking about like, is there anything where a disproportionate amount of the energy is going? Is a lot of it for synaptic vesicle release and the regulation of that? Is a lot of it for other stuff?
Yeah. So this is a subject that's a bit fraught with somewhat different opinions, but it's based on slightly different types of data and also different interpretation, just to be fully transparent about this. So one of the things, we'll get to this later, but we made this observation more than a decade ago, that how fragile synapses were metabolically, that they broke rather quickly when you cut off their fuel supply. So that's really what convinced me to keep studying it, because it was such a big effect. I felt like there was things that we might be able to make hay and understand it better because there were big effects. Now I want to separate though a little bit of confusion. Because they break so quickly, it doesn't necessarily mean that we found the most expensive thing. We found the most sensitive thing. And the analogy I like to give, if I ask, in your budget to run your life, right? Most people, when they reach a certain age, the biggest, once they're adults, is they're paying for their mortgage or their rent. Right? On the other hand, if I drop your salary by a factor of 10, I'm not going to kill you. You might become homeless. You might not be able to pay your rent. You'll scramble. What will kill you is if I stop feeding you. Right. So the food isn't necessarily the most expensive part of your budget, but it is the most sensitive part to keeping you alive.
Right.
So I use this analogy of the synapse. We know that what we've discovered for sure is an extremely sensitive point that breaks easily if we don't provide it with enough energy. The question is, who was the big consumer is a slightly different question that truthfully, we're still working on, and we think we have some pretty good ideas. A historic view, which comes largely from theoretical work, was that the brain's electrical activity is what's so expensive. Because in the same way I talked about the fact that when you put a lot of neurotransmitter in a vesicle, it costs you energy because there's a huge gradient, thousands of molecules in a tiny volume. That same logic applies just across all membranes of the plasma membrane, that there's ion gradients. That's the basis of how all cells communicate.
So I guess the idea would be, because, the whole point of a neuron is they can fire an action potential that requires these massive fluxes of sodium and potassium and so forth. And then because that creates such strong gradients, you need a lot of pumps to create those gradients.
Yeah. Now, so I would say that's largely been a theoretical estimate of how expensive the brain is. I would say that, it seems so appealing. And of course, I would say of the era, say 15 years ago, it was easy to write down estimates of like how expensive this might be. I have to say there are a bunch of unknowns, because the number of ions that actually cross the membrane to create an action potential is not necessarily that high. And it depends on, we need to know more things than we know to have an accurate number. What those calculations tend to have ignored historically was anything else, because they didn't know how to do a calculation for the synapse. They knew how to do it for excitability. And so the historic view has been the reason the brain's so expensive is because of electrical activity. I would say all the evidence we have now is that the axons themselves are not that expensive to keep running with electrical activity. It ends up being the synapses being things that are probably the things we have to pay attention to. Now, there may be exceptions, of course, and the brain is a complex organ with lots of different cells, but the synapse had been ignored as an important part of this. So we know two things. One, it's a super sensitive part that really breaks far before the ability to fire an action potential if you cut off fuel supply. The other, the data we have now coming that's not published looks like I think actually some of it is published that it wasn't so much the electrical activity. It really was all about the synapse and why it's so expensive. I wanted to separate the two concepts of expense versus sensitivity.
Right. No, I think that's an important thing to drill home because, I mean, just to kind of riff on the analogy you made, but in a different way, if you were to cut my salary in half, I would make sure to keep paying my rent. I would cut lots of other stuff, but just because those things are getting cut, that doesn't make them more important. It actually makes them, they're less important. That's why I'm cutting them. Yeah. So, but synapses are expensive, and there's a lot of them, no doubt about it. I want to talk a little bit about energy sources in the brain. I actually don't know so much about this myself, so I have a lot of I don't actually even know what the known unknowns and unknown unknowns are here, but the mitochondria can make ATP, say, from either fatty acids or from sugar, from glucose. And I've covered those kinds of topics in different ways on the podcasts on previous podcasts, but not in the context of synapse function specifically. Well, I want to start out very basic here. Is the brain, and especially in synapses, is it mostly using glucose as a fuel source? Is it using fatty acids at all? Has our picture of that started to change over time? What can you tell us about preferred fuel in the brain?
Right. So the dogma has always been that the brain largely runs on glucose. I mean, in some simple ways, it's not that glucose isn't super important, but just an example of what you learn either as preparing to go to medical school or in medical school, that it's very important to try and keep blood plasma glucose levels relatively stable. And the reason it's always given is because the brain is so hungry for glucose.
Yeah.
So you don't, and in reality, so an example I always like to give in seminars, if the sensitivity of the brain, if I gave you a shot of insulin, and I could cause your plasma level glucose to drop by just a factor of 2 for the average human, down below, let's say I crossed the 50 mgs per deciliter threshold, right? Yep. So that's about 2.8 millimolar. Most humans would begin to manifest neurological symptoms.
Okay.
Yeah. So it's within a small number of minutes.
Yeah, and that's a big drop.
It's a but it's a big drop. But you say, well, it's not much of a safety factor. It's just a factor of 2.
Yeah.
Right?
Okay.
So it is a big drop, and of course, that's unusual. I could force it to happen by giving you insulin.
Right.
And in most healthy people, you'll never get that kind of a drop. But the reason you won't is because you have a liver that will make glucose.
Right.
And so we've been endowed with organs to make sure it's kept as constant as possible given other constraints, right? So that is part of why the thinking about the brain is an obligate user of glucose. Now the question is, are there other uses? Now, one thing we strongly suspect as the field, and there's good data on this, is there is another source that we know this from therapeutics, which is for children that suffer from, particular children that suffer from intractable epilepsy. And so this what we call intractable is they're drug resistant, that they're nothing has seemed to helping them. The last last resort here is to go to a severe ketogenic diet.
Yep.
And that's quite effective at something like 40% of those intractable cases. Those children really benefit from a severe ketogenic diet. So that, of course, is under a severe ketogenic diet, you generate a lot of ketones, usually in the liver. And the thought is that those are getting the brain to be metabolized. And for some reason, there's now some bias, presumably, that's writing the imbalance of electrical inhibitory activity. Still not understood.
Yeah, so still not understood. So ketosis is effective for many forms of epilepsy like that. Sort of implicit in that whole thinking there is, okay, if you're shifting the bias towards ketones when you do a severe ketogenic diet for that particular therapeutic application, that would imply that under normal conditions for most people, maybe the brain is biased quite heavily towards glucose rather than ketones. Is that the case?
Yes, I think for ketones, that is the case. Now, I just brought up the ketones as sort of an example, which I don't consider it the same because of machineries it needs, but it is a short hydrocarbon chain, a ketone. Now, but the longer ones, which was what we tend to consider fat, and what caught my attention is this sort of extreme sensitivity of the brain to interrupting fuel supply. So I gave you the example of insulin injection, as as an Gedanken experiment, where if I did that to you, I'm pretty sure I knew what would happen. But in addition, if you there are other ways to cut off your fuel supply. You could simply get strangled, and that'll cut off circulation. The same things happen rather quickly.
Right.
And so the question is, so we know the brain really always needs a constant fuel supply.
From outside the brain, right? I'm sure the brain does have the ability to make stuff inside of itself, but what you're saying is you can't go that long without pumping stuff into the brain through the blood from outside.
So it probably is a bit of a hybrid vehicle, though.
Okay.
Which is it isn't purely running on the traditional gasoline of glucose,
right?
Such that there may be other systems that can run in parallel, and it's just that we don't quite know the rules of our hybrid vehicle yet, right? So, if you have a nice hybrid, Toyota Prius or something, well, you're used to using it, and you know how far you can get with its recharging system, etc. I'd say we don't know those rules yet, but the evidence is accumulating that we really are a hybrid type of fueling system. And I would say the example I wanted to give is muscle is kind of like the brain, which is muscle is also very expensive to contract, and it needs fuel to do this job. And it has lots of things. If you go out and run a marathon, those muscles are going to do their darndest to make sure it gets all the fuel they need, and that it soups up the machinery to use that fuel, both to bring it in and to use it well. And muscle is famously adaptable. It's very flexible metabolically. It will use glucose, but it too is a hybrid kind of machine.
Right.
And one of the fuels it famously can use is fat. And I think I would say a starting mystery is, here's an expensive machine, the muscles in our body, and they're doing everything they can to make sure they can be adaptable for what kinds of fuel they have. And the dogma that had been around for 90 years was that, neurons don't have access to one of the pieces of fuel that muscles do, which is fat. And it seemed a bit of a conundrum that why would the brain have given up, since they're also very sensitive to all these things. They're very expensive. It seems like a good fuel source. So muscles have decided to use this. And I would say the important clue that was sort of where the light bulb went off for some of our work is that if you can take a picture of muscle, usually this is done in some kind of biopsy or ex vivo preparation, it's only in resting muscle that you see clear evidence of the fat depot, which are these lipid droplets those are a fuel depot of fat molecules that are also kept inside. They're not like a synaptic vesicle. They're actually filled with a kind of fat. And those are easily visible depending on how you want to look for them, say in electron microscopy. But it's not true in exercised muscle. They're gone. Or they're much smaller in size.
Which implies that they were used.
Which implies that, it's because they're using it. Now you go back to the brain, and there's a background here that dates back 90 years, but one of the pieces of evidence that I would say is the most current one that people like to think is, we don't think the brain uses fat because we never see those fuel depots.
Okay. I mean, that makes good common sense. We don't see those. It makes good common
sense, but there's two interpretations of that, which is one is that, is it because you never use them, or that
because you always use them?
Always use them.
Oh, okay. Interesting. Okay, so these lipid droplets, let's just unpack exactly what those are. If I'm hearing you correctly, right? So it's not like a vesicle where it's a membrane that's surrounding chemicals. This is literally a bubble of fat.
Well, yeah, it's oddly enough, it's a little bit like a membrane because so most traditional membranes have two layers of these things called phospholipids, which is a fat molecule with a charged group on top. And that they like to sandwich together so the charges are separated, but the middle is very oily. And that's how all membranes that you think of are made, right? The ones of synaptic vesicles, the one on the plasma membrane, all the organelles in our cells, etc. Lipid droplets differ a little bit, but they have a monolayer of phospholipid.
I see.
And that's it. And then inside is just jammed full of usually one of two types of molecules. Just to be completely generic, cholesterol typically is found inside these. But then a thing called a triglyceride, which is basically a kind of fat that has 3 carbon chains. Most of the ones that make up traditional membranes are 2 carbon chains.
Right.
Triglycerides, as the name kind of implies, has 3.
So they're basically holding a greater density of fat.
Yes.
Yeah.
Exactly right.
So let's take those one at a time. So you can have these lipid droplets within cells. You said in muscles, we often see this in resting muscle. We don't see it in muscle that's being used because presumably those fats are being used for the muscle moving. In the brain, we don't tend to see these things as readily, but we didn't know historically, does this mean they're not there because they're not being used, or we're not seeing them because they're always being used and turning over and getting used up? When would a lipid droplet, in what context are lipid droplets filled with cholesterol? What would that cholesterol actually be used for?
We're now skating over things which I'm not an expert on.
Okay.
So cholesterol has lots of roles. It may have no real purpose in these things. It may be purely a consequence of how it's built. It's because lipid droplets are all formed by emerging from the endoplasmic reticulum.
Okay.
And the endoplasmic reticulum membranes typically have quite a bit of cholesterol. So It could be just an epiphenomena that when it's built this way, as you build this thing, the cholesterol comes along for the ride.
It's built from pieces that already contain lots of cholesterol.
Yeah. So that may, as I said, it's a bit of a soft answer because I don't really know if it has a true role.
Yeah.
If we think of this as a fuel depot, cholesterol is not really an oxidizable fuel.
Right.
It plays many other roles as precursors for steroid hormones or things that are related, or it changes properties of membranes or the proteins in those membranes. So it has lots of fascinating and important roles, but not typically one as a carbon source for combustion.
Right. So a priori, that's part of why I was asking. It's like, why would there be cholesterol there? So it could be an epiphenomenon. It could be something else. We just don't know yet. Right. But they're made in the endoplasmic reticulum. Let's just give people a very short explanation for what the ER is normally associated with, functionally speaking. Sure.
So I would say it's funny. It's a bit of an underappreciated organelle within neuroscience. It is a giant stretch of membrane because it is an internal organelle that I would say its two famous functions are depending on what part of the cell you're in. One, it is the place where a lot of proteins that have to be secreted or participate in the machinery of secretion, so which I would call in the secretory pathway, they're actually synthesized on the ER membrane. So there are ribosomes that come and dock onto the ER. It's called the rough ER. And it was called rough historically because it was studded with ribosomes, so it looked rough. But that's the place where you synthesize for the first time membrane proteins. So all ion channels, all transporters, and if anything that needs to cross the membrane is a protein.
It's like a docking station for ribosomes, and you're docking them there because these are membrane proteins. So you want to kind of make them in the membrane environment they're going to end up working in.
That's right. And so of course, that's typically all within the sort of cell body region is where you find rough ER.
Yeah.
There may be some exceptions in neurons to that too. But the reality is that ER is a contiguous organelle that then also extends through the entire distance of the neuron, all the way through the dendrite. It is also all the way out to the axon to the very end. And the traditional role it's been associated with is as a place to store calcium ions.
Yep.
So, and I'll give you the better understood example, which is muscle contraction. Of course, is famously driven biochemically by calcium elevations in the cytosol to trigger all the machinery involved in contracting actin and myosin together. But the big source of calcium is actually not really the outside world for muscle.
It's the internal
inside world. And in muscle, it's got a different name, but it's very related. It's called the sarcoplasmic reticulum.
Yep.
And it's famously where you store the calcium that you use to then do a lot of things that you need calcium for. Now, so in neurons, calcium is also being stored inside the endoplasmic reticulum. I would say it's since we don't have a contractile machinery like we do in muscle. We think of the big thing as secretion, letting out neurotransmitter. Its role there is much more subtle. I would say it's specialized, where the role of calcium in the endoplasmic reticulum may be playing, in some cases, a modulatory role more than anything. It's not considered the essential source of calcium that triggers synaptic vesicles to fuse. It seems to have very important roles. I would say it's still a frontier area because it's not been studied nearly as much as the muscle one, but there's a lot of progress that's being made. It's fascinating. And one of the roles it has that we know from some beautiful work of the last dozen years or so, is that this is the place that you build. Well, first of all, the endoplasmic is the place where you synthesize fat. All membrane lipids are made in the endoplasmic reticulum, practically.
Okay.
And
that's key. So the membrane of the cell this is where it comes from.
Oddly enough, that's where you put the machinery that makes the membrane. Of course, it's made of membrane itself, but it does all lipid synthesis. And one of the types of lipids it synthesizes are triglycerides.
Yeah.
And so the idea that's emerged from the last 15 years or so is that as you build triglycerides, they begin to form, because they're neutral lipids, they tend to form in the middle of the sort of the bilayer of the bilayer of the endoplasm is regular membrane. It's bounded by a bilayer, and in the middle of that bilayer, the triglycerides, because they're not charged, they're neutral, they tend to like form what's called a little lens in the middle, and suddenly this little thing bubbles out and emerges from the endoplasmic reticulum as a lipid droplet. Now, there is protein machinery that gets associated with it, the surface of it goes, and it carries one monolayer of phospholipid. So that's where it's born, so to speak. But they're not the only fats that are being made there. Yeah.
So endoplasmic reticulum, key organelle of cells. It's this blob-like thing, and part of the blob, usually closer to the nucleus, is what we call the rough ER. You get a lot of ribosomes, the things that make proteins, they dock onto it, and it's sort of like a docking manufacturing station for membrane proteins. There's another part of the ER that's contiguous with that extends to other parts of the cell.
That's called the smooth ER.
Yep, that's the smooth ER. There aren't ribosomes docking there. But the ER is a place where the membrane, fats of the cell are actually being made. The cell membrane kind of originates from the ER, and these bubbles of fat called lipid droplets also emerge from the endoplasmic
That's
reticulum.
Correct, yeah.
And so that's what we were getting to. Sometimes those bubbles, those lipid droplets, contain a lot of cholesterol. We don't know why that is exactly yet. And sometimes they contain a lot of these triglycerides, which is one glycerol backbone stapled to 3 fatty acids. Yes. So that can be that's a nice little pool of energy that you can put in these lipid droplets, a bunch of triglycerides, which could, if the cell wanted to, then be oxidized and used for energy. So what can you start telling us when we go in this direction? What are these lipid droplets used for from an energetic perspective at the synapse?
Right. So we published this paper recently. It was just a couple of weeks ago, but I would say up until, for most of the world, the view was the neurons really don't use lipid droplets because they never see them. So what is there to discuss if they're not there?
Yeah.
Right. And I was worried about this, the whether we were interpreting things correctly, because of the statement that the absence of something doesn't mean it's never used. It just means that it's steady state and may never build up. Right? But then there was very old, quite lovely data, but it's from 90 years ago, and it was sort of classic biochemistry of saying, let's find out what tissues use fat as fuel. And so here's the classic experiment. You have to rig up an experiment. Can you detect whether respiration would happen? So respiration is the process in mitochondria, right, of doing oxidation. And along the way, it uses up oxygen and creates CO2. And so this very clever experiment from the 1920s, early 1930s, basically set up what's called a differential manometer. 2 flasks, right, connected by a tube filled with liquid. You put a chunk of tissue in one of the flasks, and actually you put in a little alkali scrubber to take away the CO2, much like Apollo 13 had to do to get rid of the CO2. So you put it there, but then it has oxygen, and then you give it a fuel, and then you simply ask over time whether there's a pressure difference that develops because you're using up the oxygen on one side. And that's the experiment that was done. In fact, it was a famous experiment because that's how the Warburg effect was discovered in part, was using a machine just like this. That's what Warburg used for this in the 20s. So this was deployed and said, okay, well, let's take other types of tissue and ask, are they doing respiration? And so they took muscle and they chopped it up into little bits and put it in the flask and gave it a piece of fat. It was more like the fat they used was butyric acid, which is the major fat in butter. Not particularly long chain, but they gave it that and said, the muscle is breathing. It's using up carbon dioxide, using up oxygen, or producing carbon dioxide and using up oxygen. They did the same thing with cut up liver and found the same thing. And then they did the same thing with brain. But it was dissociated, sort of chopped up brain. and they couldn't really detect anything significant going on. And so that was the first one that sort of, if you go deep back into literature, that's where it began. But now we know after 100 more years of efforts to study brains that chopping up a brain does not leave it in terribly functional, right? Well, individual muscle cells can still contract. They may not do it as well when they're not in the tissue. Individual hepatocytes can still do their job. But neurons really do, because of their architecture, they don't do well when you chop things up because you tend to lose the dendrites and the axons. And so it was unclear what physical state was left for these guys to do anything.
Right. So I think an important thing I want to emphasize here for people is this is sort of a classic trade-off in science, but in neurobiology, right? Sometimes you want to remove a piece of tissue from the brain of an organism in the form of a slice, or you want to grow cells in a dish because it's much easier to study them. You can go in much more depth and detail when you do study them, but at the same time, you've created a completely new context, and things simply won't.
And I'm not critical of this experiment from 1933. I think it's a beautiful design.
Yeah.
It's just it comes with a price. They didn't quite understand all the things they were trading off. And honestly, most of us never do understand all the things we're trading off as we're doing them.
Right.
And so that's how science evolves.
But the simple fact is, when you're doing experimental science, you just can't look inside the brain and understand things in their context at the level of detail you want to in most cases. So you have to make these experimental choices of creating this new kind of preparation to dig deep into detail, but also divorcing the whole system from the one it actually works in an animal.
Yep, no, absolutely. Now you fast forward maybe another 15 years, they had, or 20 years, they had a slightly more sensitive method. The intervening time, radioisotopes became much more readily available for biochemists.
Mm-hmm.
And now you could label fat, you could make a fat that had a radioactive carbon, and instead of detecting a pressure difference, you could say, I'm going to look for radioactive carbon dioxide. So if I gave it a radioactive fat and I detect carbon dioxide, that's another definition that I am undergoing combustion and respiration. So now it's much more sensitive. And so basically but the same experiment in reality was just carried out with a much more sensitive approach 20 years later. And they concluded they can now detect a little bit of stuff going on in this minced up brain tissue, much less specific activity than the other tissues they looked at. So I would say it shifted only. It's 1933, you would say the dogma was the brain does not burn fat. 1951, some fat.
Okay.
And then fast forward another 35 years, a sort of a version of this, a little bit more modern, where they said, well, since there's some, maybe it depends on the cell type. Yeah. So then the attempt was to say, let's isolate different cell types of the brain, keep them alive in a culture dish for a few days, a week or something. But then they did the same experiment. They scraped the cells off a dish, gave it a fat that was radioactive, and asked, are we detecting radioactive carbon dioxide from astrocytes, oligodendrocytes, or neurons? Those are the three types studied. This is circa mid-80s. And that experiment concluded only the astrocytes were burning fat. No detect bacteria from either oligodendrocytes or neurons. But I'll argue there's two flaws that have historically plagued this. One is a bit of an assumption that if you give fat to a cell, is it readily available for combustion? How long will it take for that to be usable?
Right.
And the second is, have you left the cell doing, purely the ability to do what the cell is supposed to do. And I will argue when you scrape cells off a dish, maybe astrocytes are intact enough to do their job. Oligodendrocytes, they're kind of a super interesting architecture, much like neurons, almost certainly don't survive this scraping off a dish. Right. Neurons definitely don't. Yeah. And so I would say, unfortunately, the persistent problem sustained itself for a good 50 years of experiments.
Yeah. And this is another sort of meta lesson here, is that oftentimes what we think is true or what has the most evidence behind it might be wrong because the methods that are available to do this just
had constraints.
They have constraints. Yeah.
That's right. So we so in all honesty, that I had bought the Dogma kind of hook, line, and sinker, right? When you decide, you're running a lab, what problems you decide are worth attacking right now.
Right.
I honestly was my own background was in a different field. I wasn't steeped in all the things I just explained to you until much more recently. And it just didn't seem like, the dogma was this: brains don't do this, synapses probably don't do it either. And in reality, the first author on this paper we just published, named Mukesh Kumar, had come to me as a postdoc, and he came from the lipid droplet world, now not in neurons, in a different context. And he was curious. He was suspicious that there was more to it than people thought. And so I basically, we had a conversation. I says, okay, well, I want to be convinced that this got some meat to it. So we discussed experiments and design the first round. But the turning point for our story really was when we became aware of a paper published 11 years ago, which was nailing down the culprit of a human disease. So these are diseases, it's in a family called hereditary spastic paraplegia. It's a very big family in the sense that there are many genetic drivers who've been isolated for this. Doing some of them are involved in controlling how well the ER works, how it's shaped, all these things, lots of intriguing things. But I would say the field tends to refer to these as long axon diseases because they cause a problem with your limbs, which have these very long motor neurons.
Okay.
Right? And so it was always sort of viewed as, oh, well, these spastic paraplegias are somehow particularly challenged. When you have a mutation, that component must be involved somehow in maintenance of this long axon that has its own challenges for how do you maintain something like that.
Yeah.
And that's the view I had superficially. But this one was an unusual one. And so the particular mutation was the disease is called HSP54. 54 being hereditary spastic paraplegia, number 54. There's been almost 90 known genetic drivers. So this is a long list. But as I mentioned, the classic disease is this long axon disease, but some of them were called complex. And the complex ones are clearly also a CNS disease. And HSP54, in addition to having the long axon disease, these people have vision impairments, ataxias, and intellectual disability. Right? Okay, so there's something extra special about these guys. And then the turning point came from a paper published 11 years ago where an investigator at the Scripps Institute, Ben Cravat, who was himself a chemical biologist and had worked a lot on biochemistry of fat handling machinery, got intrigued with the enzyme, the gene encoded by HSP54, which had an equally attractive name called DDHD2. And he basically demonstrated, I would say quite conclusively, that thing is a triglyceride lipase. And it's particularly enriched in neurons.
So it's an enzyme that processes triglycerides.
Right. And so for you to use those triglycerides stored in lipid droplets all over the body. So, for example, lipid droplets are also found in adipose tissue, the things that builds up when you're fat. And you have giant lipid droplets, but those lipid droplets, no matter who you are, you can burn that fat, or at least they could be liberated from the lipid droplets. And the things that liberate them are enzymes that go to the surface of the droplets and actually begin to break the triglyceride down into fatty acid components.
So that enzymatic step needs to happen in order to take that lipid droplet and transform it into something that can then be burned for energy.
Yes, exactly right. Exactly right. And so I would say that it should have been, I don't know why it didn't resonate as much with people when I thought it was a very fascinating paper, because what they showed is that, first of all, it's just a straight experiment. They knocked this enzyme out of mice.
Yep.
And the mice have lots of problems in the brain. There's lots of other kinds of evidence of whole animal behavior that something is not going right in the brain, consistent with the fact that in humans it's bad news to have this mutated. But you can do clever experiments, which is say, let's compare the lipid profile of the brain by lipidomics. So these are analytical techniques to allow you to characterize all the types of species of lipids that are in a mouse brain.
Yep.
And so you can compare this in an animal that is missing the enzyme versus one that isn't.
Okay.
And
so what does the fat profile of the brain look like when an animal is normal and when an animal?
Right. And it was absolutely clear-cut, no ambiguity, giant accumulation of triglycerides in the animal that's missing this enzyme.
I see. So if you don't have this enzyme, you start to accumulate these lipid droplets that are packed with triglycerides.
Right. And then they had other evidence. So it's one thing to see a biochemical signature, got lots of triglycerides. They showed by electron microscopy, they look at a mouse brain. Sure enough, the neurons are chock full of lipid droplets, something you're never supposed to see.
Okay, so where did you guys take it from here?
Okay, so
yeah.
So we were on the hunt and building up our own evidence with a very reduced system that we can study, but we have developed tools over the years to study in a reduced way, neurons in a dish, but in a very accurate way to say, are they making ATP during activity? Or what are they using to make the ATP during activity? And how well can they do it? Right? And both how important are the mitochondria, how important are glucose? And we had come to this conclusion that if we force-fed neurons in a dish extra fat, then actually they would have lipid droplets, and they could use these lipid droplets as a source of fuel.
So if they're indeed using these things up all the time, and there seems to be problems when you mess with these lipases, that implies that something interesting here is maybe happening. So if you just give the brain a surplus of fat, maybe you will accumulate these lipid droplets to the extent that you can start seeing them more readily.
Yes, that's exactly. And we have been doing this when, as I said, it's a sort of lesson in science. Nobody really has a full scope of all types of science that's relevant at any given time. So you have to be ready to decide to learn a new thing, which is what happened to me when I got into this field. And then we came across this work from a decade ago about the studies from Ben Cravath, and there was one follow-up that was relevant after from his lab, but the DDHT2 was a neuron-specific triglyceride lipase, and so we started realizing that may be an interesting handle for us to take advantage.
And also the fact that this was neurons, so this was a lipase that is only expressed in neurons. That kind of hints at something.
Yeah, exactly. And then it connected it to and always an advantage in any argument you make when you can rely on human genetics. It eclipses most worries when the interest in something starts with identification of a specific thing that leads to a human disease that we know is, in this case, in the brain.
Because now no one can really argue, well, this is just some trivial
thing that
isn't going to connect.
Which it's a fair thing, because you don't ever know. You might think you found the most fascinating piece of cool machinery, but building up evidence that it's as crucial as you think it is, usually you make knockout mice, and often that still doesn't survive the test after a knockout mouse. In this case, starting from a human disease that has this specific mutation, this one thing leading to these problems. The mice have the same similar problems, I should say, not the same. That was realizing all that. We realized that we'd sort of hit pay dirt, because they had built a case for us, and now we could go in and sort of, in some ways, close the loop, and which didn't happen in the first papers, which is to say, what are these lipid droplets for?
Yeah. All right. So what happens next in the story?
Right. So we developed sort of associated tools to study, and when we took advantage of another very useful resource that Ben Cravath, who is a chemist as well, he had made an active site inhibitor of this enzyme. So this meant that we could do rapid experiments. So we could basically take neurons, block the activity of this enzyme DDHT2, and realize that even sitting in an incubator overnight with the enzyme not working, we built up lipid droplets everywhere without even feeding them fat.
I see. So now you can reversibly inhibit this enzyme instead of just doing a total knockout, and you see these lipid droplets accumulate.
They accumulate. And one of the places they accumulated, lo and behold, was at synapses in nerve terminals.
Okay.
And so suddenly now it's sort of really coming close to home. It's like, it's the thing I care about, they're building up there. And we took advantage, I would say, the pivotal thing, which was not evident, at least maybe it was evident, but it was hidden from me in Ben Cravath's paper, but it wasn't relevant to the story. It turns out his inhibitor was reversible.
Okay.
It didn't need to be for anything they did. They just wanted to show that they could. I think he made it because if you block the enzyme it's one thing to knock something out in an animal and show when it grows up, it's got all these problems. It's another thing to go, well, was that a developmental problem? Or if I could do it acutely, can I get a problem? Right? And so pharmacology can often get you pay a price, but it can give you more direct answers about some things. So he made a pharmacological tool and showed that if you gave that into an animal brain over several days, you build up lipid droplets easily visible. So it happened in an adult. You didn't have to happen while you were developing a nervous system.
Okay, so these lipases are indeed involved in something to do with the formation of these lipid droplets in synapses.
Yeah, so one of the things because we had a little bit more of both a physiology bent and a cell biology bent of synapses and neurons, we asked the question, where do you find this enzyme DDHD2? And of course, it's in lots of places, but one particular place we found it was at nerve terminals in the axon.
Okay.
It was pretty enriched there. Now, it's not the only place, but that, if you study synapses for a living and you see something's been highly enriched, it says, this is playing some special role at my favorite location, right? Mm-hmm. And so we noticed it there, and sure enough, when we blocked its activity, we grew lipid droplets at synapses, or at most of them. It was about 2/3 of them grew a visible lipid droplet. But because this was the drug that was reversible, we can then ask probably the most important question is, once you've built a lipid droplet, what can you do with it?
Right.
Right. And I'd say that's what is not so easy. If we didn't have it reversible, we couldn't ask that question. And if we, when the enzyme's working, we don't see lipid droplets.
Yeah.
So this is why it ended up being a very important tool for this study, because we could build them, then unblock the lipase and ask what happens. And I'd say there's two facets to this that were critical. One is a clever experiment that Mukesh designed, which was to put a tracer of a fluorescent fat while he built the lipid droplet. So he gave the neurons a small amount of a fluorescent fat, really just the tracer amount. And we knew that we were building lipid droplets when we blocked the enzyme in a matter of hours. So he says, okay, well, let me put in a fluorescent tracer that has a good chance of ending up in lipid droplets. It didn't have to, of course, but it did. I mean, he knew from his own background that this thing would go into lipid droplets typically. And sure enough, it did beautifully in neurons. So now we had fluorescent lipid droplets. So now we have something that's got a marker on it. And when you unblock the enzyme, you can ask, where does that fluorescence go? And what's a process of burning of fat by mitochondria involves taking up fat into the mitochondria. It has to cross the double membrane system. Yep. There's detailed machinery to bring in these oily carbon chains into things. They're special transporters for this. And so we had just relatively simple tools. Is okay, can we detect whether or not when we unblock the lipase, it ends up getting shipped into mitochondria, which would be a necessary thing if you're going to use it there to make ATP.
So if these lipid droplets are being used for energy, the lipase needs to break up the triglycerides in the big lipid droplet, and then those fatty acids that come from that need to get into the mitochondria where they can do beta oxidation to create ATP.
Exactly. That's right. And so there were a few criteria we used to convince ourselves that we were getting a bona fide signature of this. One is that when we made fluorescent lipid droplets with this tracer, if we're going to be doing beta oxidation, we should see an accumulation of the fluorescent lipid inside mitochondria after we unblock the lipase. And so that's exactly what we saw. And it provided us a very important insight, which we're still chasing, which is that the time it takes to go from the lipid droplet to the mitochondria, in this case, now the time may be a little bit artificial, because we're not controlling everything, but it took a few hours. Okay? That time scale doesn't necessarily mean anything per se. If we have ways of blocking the transport machinery that is responsible for bringing fat in, and there's a drug, for example, or you could do a knockdown, remove a piece of machinery. If you did that satisfactorily, that also prevented the fat from getting into the mitochondria. So it was relying classically on what it's supposed to rely on to do beta oxidation. But the other one, which I think to me is one of the most interesting insights for us, is that electrical activity also controlled this. So if we kept neurons so-called in the dark, which is we just silence them electrically, which you can do by blocking their sodium channels, so they just can't fire any action potentials, the lipid droplets didn't move their fat. Even in the presence of an unblocked enzyme, it didn't go in. I took this, truthfully, as a hallmark of synapses need to make energy on demand. And here's a process that is being controlled by demand.
Yes. So if you block action potentials, the bubbles don't go anywhere because presumably they're not needed to be turned into energy. What happens if you crank up the action potential frequency?
The opposite happened. They go in faster.
I see. Yep. So that's a pretty good indication that these bubbles of fat are being used to power action potential related stuff happening in the synapse.
Right. And another thing, back to the analogy I made, or I would say the glimmer that first noticed that maybe the absence of fat should not be interpreted as it being not be used, is the example in muscle, that resting muscle accumulate fat, but not working muscle.
Yeah, yeah.
What we also notice is forgetting about putting tracers in, just silencing neurons for, say, 24 hours, they build up lipid droplets on their own.
And that's by analogy, the silenced neuron is like a resting muscle.
It's exactly. So this sort of became full circle to us that maybe our brain is just like muscle. It was metabolically flexible, and it will use fat as an energy source. Yeah,
and the fact that you don't see those lipid droplets is actually an indication this type of thing is happening all the time.
That's right. Yeah.
And so what starts to happen when you think about this in the context of the whole animal? Did you do experiments where you inhibited this process, this ability of this enzyme to work on these triglycerides? Does that affect the sort of whole animal at the metabolic level or the behavioral level?
It does. Now, yes, it does. And actually, this I would say is clearly a frontier area where we have to try and be as clever as possible to begin to ask this in interesting concepts. But as a starter, I again, this is the serendipity of science. I was having a discussion with a different colleague and I found out about an experiment done literally across the street from me at Rockefeller University by a medical fellow. And all he had been doing is he had been blocking at the whole animal level the machinery to bring fat into mitochondria. The machinery is called the carnitine palmityl transferase, CPT1. So that's the first transporter to bring fat in. There's two of them you need.
You guys are working with an enzyme that kind of chops up the triglycerides and these fat bubbles. After that, they need to get into the mitochondria. Now we're talking about something that transports at that level.
Right. And this gentleman's name is Kartik Rajagopalan, and he, as I said, he's now has his own lab at UT Southwestern, and he's a physician, but he was working in a lab that was sort of interested in other aspects of how the brain senses energy needs as opposed to the details of how it gets the energy. It's more like circuits involved in feeding and things like this. And so they had done this experiment where they had injected a blocker of this transfer. Now, that blocker would work, all cells that use fat use this machinery. And so his experiment could have been blocking systemically all the transporters, so anywhere. But what he discovered is that, now he did inject it, he injected it, but he discovered that under this particular condition where he had food restricted the animals, which is the kind of thing that lab would have done as a typical experimental paradigm, injected this in, drove the animals into a very strange state called a torpor state. So torpor, and I'm not an expert on this, but it's sort of, I view it as an adaptive mechanism that's probably metabolically protective to say, like, and it's maybe related to hibernation, for example, where you begin to shut things down and you go like, something is scarce here. And so maybe an extreme thing. I'll give you an example. About 4 years ago, Michael Greenberg's lab at Harvard published an influential paper where a very sort of cool way that people break circuits now is you take advantage of the fact that when certain neurons begin to turn on, there's a gene marker that's easily seen called CFOS. Right? And so you can go, let's put animals into, and you can drive this torpor state in animals with a combination of various metabolic stresses. One is you can deprive them of food. If you do that and you cool the animals down, they kind of freak out and they go into a torpor state. So the Greenberg lab had used this as a leverage point to say, what are the brain cells driving this torpor state? And they discovered, not too surprisingly, there was a set of hypothalamic neurons that are the ones that are getting turned on. Then they use other tools to show if we turn those neurons on, we can drive a torpor state. So
there's neurons in the hypothalamus that turn on torpor. And those neurons can presumably be activated by things in the environment that are indicating the animal should basically conserve energy.
Yep. And so I had heard about this experiment that Kartik had done, just sort of indirectly. But as soon as I heard about it, I wrote him an email and he came over and we chatted about why I thought his stuff was interesting in our context. And I said, an interesting experiment is to not use a systemic-wide blocker of all transporters. But how about, I bet the same, I said, I'll gamble that something related might happen if you block things. But now it'll only be blocking a machinery that is generating the fatty acids you needed for that transporter, but it'll only be doing it in neurons. And so that's the experiment we came up with. And Kartik did these experiments, actually largely after he had moved to his own lab now. He was sort of setting up. So he was very gracious because he was spending precious startup time with a fledgling lab with a couple of technicians to do experiments that they're not the kind of things that we do. And lo and behold, he discovered it was almost identical. That was slightly more severe with this drug. Now, that might have to do with potency as much as anything.
So let me get this straight. So basically, the idea here is you inhibit the ability of neurons only to utilize these fats for energy, and that makes them go into a state of torpor.
Yes.
Which is akin to hibernation, sort of. It's like hypometabolic state.
For my own superficial reading, that's what it is. It's sort of
body temperature drops.
Sort of use this as it's a combination of metabolic stresses will tell you it's time to start preserving things.
Right.
And so we interpreted this as, well, we gave them one stress, which is food restriction. And lo and behold, the inability to burn fat, even though you probably have, your glucose levels may be normal.
Right.
Is the brain is reacting like, this is a metabolic stress I can't take.
And this starts to bring us back to what we were talking about earlier, right? Which is, the dogma for a long time has been the brain is all about glucose primarily. And sure enough, like the brain uses a lot of glucose, and glucose is very important.
No doubt, yeah.
But what you're saying is that if you inhibit the ability of neurons to utilize this fat source of energy, even though the animals are not food restricted and they presumably have all the glucose they could ever want.
Well, actually, they were food restricted, but my guess is the glucose levels up. Actually, we don't have measurements. I'd have to go back to the literature whether they know for this food restriction, but our bodies try to do a good job at keeping glucose levels relatively constant. That's what our liver will make glucose out of these stuff. We didn't make measurements like this for these conditions. Kartik's lab may know those numbers about how much it had fluctuated. So I don't want to be, and what we really don't know is whether it had changed in the brain.
Right.
I want to, it's very important to point out that these things I mentioned about plasma glucose and how sensitive we are, we actually don't know mostly what the concentrations are anywhere in the brain during these things.
I see.
So that's an open question right now.
Okay. But the empirical result here is you induce torpor by messing with the brain's ability to utilize these fat droplets.
Yes, that's exactly right.
So what do you think how you guys interpret this, in general? What do you think this actually means?
So I right now I'll take it as, in some ways it was a disappointing result. It was a quick, easy readout, but it was an overriding readout that the animal went into torpor. So you're not going to be able to study other brain circuits when this thing goes into torpor. So it just meant it was a master switch in some ways that said, okay, too bad. But because this lipase is it's not just in hypothalamic, truthfully, we don't know what neurons mediated this torpor response in the sense that which ones realize there's not enough energy. That's a circuit question that we didn't probe, right? But the fact is this lipase is in hippocampal neurons. It's all over the place, right? So presumably that it means, to me, the big question is, it is always a source of energy. It's always there. Now, it may be particularly when times are scarce, but I would say the fact that we never see lipid droplets means that we're probably constantly burning through them at some rate. And to me, that's a starting point for discovery now going forward, because we didn't know it was there. Now we know it's there, but now we would like to figure out, is it important for certain types of function outside of the hypothalamus? And what are the particular conditions that you'll see it more easily, the consequences of you're not being able to use it. So those are two of the big things that we'd like to figure out.
Yeah. And so, a lot of people, when they start to learn about metabolism and the metabolic states, you start to get into, you learn about ketosis and you're like, oh, okay, like there's different metabolic states the body can be in. We can be burning sugar for energy, we can burn fat for energy. It's too simplistic to think about it as we're either burning sugar for energy or fat for energy, and it's one or the other. It's probably always some mixture of both.
Yeah, I do think, I like the analogy that it's a hybrid, where our brains are a hybrid vehicle.
Yeah.
And although maybe in a modern hybrid, car engine, it is picking one of the two sources it has
at any given moment.
At any given part. I think in reality, there's more analog version where it's a bit of a mix. The might be controlled in ways we don't understand.
Right. Is it
and
80,
maybe
20,
one
is it
extreme
50,
is all or none. But I suspect it's a continuum in between the two.
Yeah. I see. Interesting. And so, what else are you guys working on today that ties into the story? Just the general idea that synapses are in part powering themselves by using fats for energy.
Yeah, so I would say, I mean, this is kind of emerging. We're not that far along yet. There's a few things. One is sort of very, you might say, very nerdy detail of molecular machines, right? Which is, we don't know the root of when you free a fatty acid from the lipid droplet, say surface, I assume, because that's where the enzyme is working. How does it find its way to the mitochondria? Because fats don't like to they don't float easily through the cytoplasm. And so we suspect there is a conduit of things that will channel it to the surface. And so we're working on candidates of those that understand these are machineries known to handle lipid flow at contact sites between organelles. And so we suspect there may be one that's important for this. And so that's one of the directions we're going in. I'd say that's drilling down to details of molecular machinery. Another one is we will be beginning experiments in the coming months of, instead of using the drug, which we can't control other than globally, right, is to use molecular tools to take DDHT2 out in specific types of neurons, and then ask in a whole animal context of where you might be setting up your specific favorite thing, right? It could be, working memory or something like this, or neuroprotection to a disease. You could ask, was it playing a role in that kind of paradigm? Where we have to pay the price, because this will be a genetic manipulation, it won't be as acute. But we don't have a simple way right now to do acute things that aren't global.
And so you did these experiments where you inhibit the ability of neurons to utilize these lipid droplets. It induces torpor in the animal. Torpor, as we said, it's a sort of whole body, whole animal state where, everything's, your body temperature drops, the animal's not doing as much. And it makes sense, because if you can't utilize all the energy you need to do stuff, the brain is telling you to stop doing so much stuff. What does that mean about how do you start to think about what might be inhibited at the behavioral level by the inability to use these fat stores? Are there experiments you guys can do where maybe the animal doesn't go into torpor, but you can start to tease out, what exactly are the behaviors and the circuits in the awake behaving animal that are really reliant on these lipid droplets for energies? Is that something on the horizon? It's
sort of not really in my personal wheelhouse. I think it's interesting, but to do that kind of level of circuit cracking, how successful it will be is a bit hard. Again, the main tool, as it's a bit blunt, right, which is a drug that blocks the enzyme everywhere.
Yeah.
Right? And everywhere in the brain. And so that's not great. You wish you could go in more cleverly and go, let me just take it out in the hypothalamic neurons. And so, were that the ones that needed it? And maybe you discover, they didn't, that's not the ones that cared, it's somebody else. But those tools work on different timescales. So, but I have to say, because of those complexities, I don't anticipate that is something that we'll be going into directly. Instead, I'm more encouraged by trying to do experiments with collaborators, honestly. Let's move away from the hypothalamus and instead take circuits that we think are engaging, that are energy demanding, and that might impact performance. I'll give you an example, although it's not necessarily about fat. It's one I'm hoping to convince a colleague to engage in. So this I find fascinating. This is published maybe 30 years ago, but there's lots of data. So if you take a room of 100 people, 2 hours after lunch, just to pick a particular metabolic time of day, and I give half the people in the room an oral shot of glucose, and the other half I'll give them an oral shot of artificial sweetener. Then I'll give them 25 words to memorize, and then I'll test the recall ability of those 25 words. So the people that got the glucose do twice as well, and that's not a small effect. And honestly, this has been studied in lots of contexts with kids before tests, that fueling the brain really does tend to help with cognitive tasks, right? And so in that sense, like if this is an important fuel source for cognitive tasks, so we don't know that it is, right? We suspect it might be. So we would like to build a case that maybe it is playing a role in much more than just the hypothalamic neurons. As cool as that system is, but because it's sort of a sledgehammer what the hypothalamic neurons ended up doing, I would rather do a more surgical strike and see, can we build a case for this playing roles in other types of decision-making, cognitive behavior and things. But as I said, because we're much more of a molecular cellular physiology lab, we will collaborate with people, sort of motivate them along those lines.
Yeah. Is there anything that you want to reiterate for people, or any final thoughts you want to leave them with about this field or about anything that we spoke on today?
Yeah, I mean, there's one big frontier for us, which I do not understand at all with respect to fat burning in the brain, which was, even though we went through the cell biology that where you make lipid droplets, it emerges from the endoplasmic reticulum as a little bubble that you can use. In the end, someone's got to be providing the carbon source to make fat too.
Yep.
And we know that we could do this in a dish artificially by giving it fat molecules, and they will make lipid droplets out of them because we've given an excess of what they need. So we don't understand, now that we know the brain must be burning fat maybe all the time, we don't know what the source is, ultimately where it was getting the fat to make the lipid droplets. And I would say that is a big question. One of the reasons people kind of discard is they didn't think that the standard way to ship fat in the body are through lipoprotein particles.
Yeah.
They vary in size, immediately upon digestion, fat gets sorted in things called chylomicrons, but there's LDLs, VLDLs, there's all kinds. The brain has lipoprotein particles as well. Largely, those come out of astrocytes and microglial cells, right? And famously, one of a very popular risk factor for Alzheimer's is in a protein that binds to lipoprotein particles, ApoE4 or ApoE, and the risk allele is ApoE4. I don't really understand. I don't think anyone has a clear idea precisely of what's going wrong when you have that allele. My favorite thing viewing this is, so maybe it's actually normally involved in helping to ship fat from that cell population to neurons to use
maybe
it's
an energy problem.
Yeah.
Interesting.
Yeah, so I think, truthfully, the energy problem is going to rear its head in lots of neurological diseases, epilepsies, neurodegeneration in particular. And I would say that to leave everyone with one note is that the biggest risk factor for neurodegeneration is the aging process. Mm-hmm. And outside of the brain, we know that the aging process leads to a degradation in fuel delivery and efficacy of fuel generation and fuel use. Yeah. And there's no reason to think the brain has escaped this part of the aging process. So I think they're going to be very closely intertwined, neurodegenerative diseases and sort of brain energy metabolism.
Well, Professor Timothy Ryan, thank you very much for your time. Fascinating work, and I look forward to following along what you guys come up with as we
very nice
forward.
To meet you, Nick. Thank you.
Thank you.
Take care.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.